Mitochondrion occupies a central position in the energetics of the cell. It is popularly known as the powerhouse of the cell because the ATP required for the various chemical reactions are generated inside this mitochondrion. This ATP is generated by substrate level phosphorylation and also by oxidative phosphorylation. The substrates required NADH plus H plus and FADH2 are in most cases generated inside the mitochondrion itself by means of the Krebs cycle and also by means of the link reaction, which is the conversion of pyruvate into acetyl coenzyme A. And also you know that in glycolysis NADH plus H plus is generated in the cytoplasm. It is transported into the mitochondrion for the generation of ATP. Therefore, the energy generation process inside the mitochondrion depends upon a constant supply of raw materials either generated there itself or supplied from outside the mitochondrion. And also in this process, oxygen is a raw material and carbon dioxide and water are generated inside the mitochondrion. You have studied that carbon dioxide is generated from the citric acid cycle while the metabolic water is generated as a result of the electron transport chain. All components of the electron transport chain responsible for the generation of ATP are attached with the inner membrane of the mitochondrion. According to the chemiosmotic hypothesis proposed by Dr. Peter Mitchell, generation of ATP basically depends upon a proton gradient, a difference in concentration of protons on either side of the inner membrane of the mitochondrion. Look at the diagram here. Inside the matrix, there is a very low concentration of protons, which means a higher pH. While across that membrane, in the intermembrane space, there is a very high concentration of protons, that means a very low pH. What's the function of the electron transport chain that we are going to study? To maintain a concentration difference, which means a concentration gradient of protons across the inner membrane. So as long as the electron flow is working through the carriers of electrons, there will be a proton gradient across the inner membrane of the mitochondrion. And as long as this proton gradient is existing, it will lead to the generation of ATP. The NADH plus H plus and the FADH2 molecules we talked about have only one function. They are the source of electrons and protons. The electrons being donated by them are transferred through a series of carriers which are organized along the inner membrane of the mitochondrion. When a member of the electron transport chain is accepting electron, it is reduced. And when it is donating electron to the next member, it is oxidized. So this is basically a series of redox reactions. While the electrons are passing through some of these members, they are pumping protons from the matrix of the mitochondrion towards the inner membrane space, P side of the mitochondrion. For our convenience of study, these electron carriers in the chain are categorized into 4 complexes. You can see it in the figure, complex 1, 2, 3 and 4. Of these 3 complexes, complex 1, 3 and 4 are acting as proton pump. They are pumping protons from the matrix towards the intermembrane space. It is this pumping that is responsible for maintaining the proton gradient across the inner membrane of the mitochondrion. So as long as the electrons are flowing, the proton pumps will pump protons and proton gradient will be maintained. Here is the detailed structure of the components of the electron transport chain. Complex 1 consists of enzyme-linked FMN, flavin mononucleotide, and also iron-sulfur centers. Complex 1, that enzyme is NADH coenzyme Q oxidoreductase, that is accepting electrons and proton from NADH. And this electron is later given to coenzyme Q. Complex 2 consists of succinate coenzyme Q oxidoreductase enzyme, which was taking part in the citric acid cycle and responsible for the production of FADH2 from FAD. This complex also consists of iron sulfur centers. Electrons are given to coenzyme Q. From coenzyme Q, these electrons are given to complex 3, which consists of iron sulfur centers and cytochrome C1. From this complex 3, electrons are passed on to cytochrome C. It is a mobile carrier of electrons. And from there to complex 4. Complex 4 consists of cytochrome A, copper ions, cytochrome A3, etc. Finally, from this complex 4, the electrons are accepted by molecular oxygen, resulting in the production of metabolic water. NADH releases 2 electrons and 2 protons. The 2 electrons are donated to complex 1 through the FMN and iron sulfur center it is passing, and finally reaches coenzyme Q. At the same time, the 2 protons are released into the matrix of the mitochondrion. From there, they are taken to this quinone to form QH2. Quinone is actually a hydrogen carrier. It accepts electron from complex 1 and protons from the matrix of the mitochondrion. Coenzyme Q is receiving electrons from 4 sources. One of them is the NADH plus H plus we discussed as in the case of complex 1. The remaining 3 are FADH2 molecules. As is shown here, the complex 2 is receiving the FADH2 molecules produced during citric acid cycle. That means when the succinate is acted upon by succinate dehydrogenase enzyme. That is one of the sources of FADH2. The second source is when glycerol 3-phosphate is acted upon by glycerol 3-phosphate dehydrogenase in the cytosolic site. FADH2 is produced and that is reaching coenzyme Q. And in the final case, when the fatty acids are broken down, FADH2 is released. Those electrons are also reaching coenzyme Q. Complex 2 contains succinate dehydrogenase. As you know, this enzyme is taking part in the citric acid cycle also. Therefore, a part of this enzyme is anchored to the membrane of the mitochondrion, while another part is projecting into the matrix of the mitochondrion. As you know, it is accepting FAD as the coenzyme. It is converted into FADH2. Its electrons are then taken and passed over to coenzyme Q. How the quinone becomes quinol? Q becomes QH2. It can accept a total of 2 hydrogen atoms, which means 2 protons and 2 electrons. In the first step, the ubiquinone accepts 1 proton and 1 electron to become semi-quinone radical. In the second step, it accepts 1 more proton and electron to become QH2 ubiquinol. So totally 2 protons and 2 electrons are used to convert ubiquinone into ubiquinol, Q into QH2. The Q cycle consists of two halves, first half and second half. Look at the first diagram. The first half is beginning with a QH2 molecule. In the previous slide you found how the Q becomes QH2 by accepting electrons and protons from the NAD or from the FADH2. Now the QH2 interacts with a cytochrome C1 and the QH2 is releasing 2 electrons and 2 protons. You can see in the first diagram the 2 protons are released into the intermembrane space. What about the 2 electrons? One of the electrons is given to a Q. It became semi-quinone radical, while the second electron is passed over to cytochrome C1 through iron sulfur center. You can see the net equation below. Now, what about the second half? Here also there is the QH2. The QH2 interacts with the semi-quinone radical formed in the first half and also with 2 protons taken from the matrix side of the mitochondrion and also with the cytochrome C1. And what's the result? QH2 is remaining there while 2 protons are pumped towards the intermembrane space while cytochrome C1 is accepting the electron from the semi-quinone radical. So cytochrome C1 is reduced and the semi-quinone radical is now quinone. The net equation is given at the bottom of this slide and these things are explained in the next slide also. Complex 3 is attached with Coenzyme Q and this complex 3 is accepting electrons from Coenzyme Q and is thereafter donating the electrons to cytochrome C. Complex 4 has two functions. Function 1, it is pumping protons from the matrix towards the intermembrane space. 4 protons. Function 2, it is accepting another set of 4 protons and a set of 4 electrons from cytochrome C and combined with 1 molecule of oxygen forming 2 molecules of water. This is the metabolic water and it is formed on the matrix side of the mitochondrion. Let us look at the chemiosmotic mechanism for the formation and releasing of ATP proposed by Dr. Peter Mitchell. As we have seen earlier, the protons are pumped from the matrix of the mitochondrion towards the intermembrane space. Now, there are so many protons with the positive charges on the intermembrane space, while there are very little protons towards the matrix side. We say the matrix side has a net negative charge with respect to the intermembrane space. This situation is resulting in the synthesis of ATP. Point 1, there is a very high pH inside the matrix, which means the pH is alkaline in the matrix. And point 2, the inside of the membrane is highly negative with respect to the outside of the membrane, that means the intermembrane space. Under these conditions, ATP is synthesized. In the intermembrane space, there are so many positive charges and you know that similar charges repel each other. So the protons want to move away from there. The only space available is the F0 base of the F0 F1 particle or the ATP synthase. So the protons are diffusing through the F0 base from the intermembrane space towards the matrix. This movement of protons results in the release of ATP from the ATP synthase molecule. In this picture, we can see the intermembrane space with a large number of protons, and there is a matrix side with very little protons. Matrix of the mitochondria. And you can see that the electron transport chain is pumping protons from the matrix side to the intermembrane space. That means in the intermembrane space now there are so many positive charges, and you have studied earlier that positive charges, or we say similar charges, are repelling each other. So there is a large amount of repulsive force here. We say proton motive force, PMF, proton motive force. And in this figure, you can see here is the F0, F1 particle, also called ATP synthase. Through this F0, F1 particle, protons are diffusing, where from the intermembrane space into the matrix of the mitochondria. So protons are diffusing, and this diffusion of the protons, this movement of the protons from the intermembrane space to the matrix through this F0 particle is leading to the releasing of synthesized ATP. Actually, ADP is combined with the phosphate forming ATP. That ATP is remaining here with this F1 particle. When the protons are moving out, it is resulting in the releasing of the ATP. And if you read some other textbooks, you may understand that there is an error in this figure. In this figure, there is an error. Actually, the proton channel is not through the F1 particle, it is only through the F0 particle. If you see some other textbooks, you will understand it easily. So, whenever protons are moving from the intermembrane space towards the matrix, it is resulting in the release of ATP, or we may simply say it is resulting in the synthesis of ATP. And again, the protons are being pumped from the matrix towards the intermembrane space. So from the intermembrane space, actually protons are moving down an electrochemical gradient from the region of a higher concentration to the region of lower concentration. This is the picture of ATP synthase in the mitochondria. You see, this is the region which is responsible for the synthesis of ATP. You see, just count here how many subunits are there. And this is the region. Can you see here in the red colored? Is it red or yeah? The red colored proton line can be seen. The pathway through which protons are just moving out. Remember, the proton channel is only in this F0 base, not in the F1 head. This is a diagram of the mechanism showing the synthesis of ATP. First of all, you read what is written there in that slide and go through it very carefully. It is actually showing the mechanism of the formation and the releasing of ATP. How is the ATP being formed? By the reaction between ADP and inorganic phosphate. Okay, and how they are released, how is the releasing and formation of ATP coupled with the movement of protons, etc. You can see from this slide very clearly. This slide is explaining as a summary the major factors associated with this formation of ATP. Can you just assume from which textbook this is taken? This is actually from a very famous textbook, this paragraph or this part of the page. Just assume from where is it taken. The ATP made inside the mitochondrion is used for running biochemical reactions within the mitochondrion and also outside the mitochondrion. For the second purpose, the ATP is to be transported out across the membranes of the mitochondrion. For that, there are the transport proteins present on the membranes. NADH plus H plus is formed in the cytoplasm during glycolysis. And this NADH should be transported into the mitochondrion to take part in electron transport chain to generate ATP. How is this taking place? In the case of liver, kidney and heart, there is the pathway called malate aspartate shuttle. In this pathway, look at the step 1, oxaloacetate in the cytoplasm combines with NADH plus H plus and become malate. This malate in step 2 is transported across the membrane of the mitochondrion into the matrix. Inside the matrix, the malate is releasing NADH plus H plus and becomes oxaloacetate in step 3. This NADH plus H plus is used for generation of ATP by means of the electron transport chain. The oxaloacetate in turn becomes aspartate and is transported out of the mitochondrial membrane into the cytoplasm by means of step 5, and the reaction is repeated. Glycerol phosphate shuttle also operates in the other tissues to transport the NADH molecules of the cytoplasm into the mitochondrial matrix. In this case, dihydroxyacetone phosphate accepts NADH plus H plus molecule and becomes glycerol 3 phosphate. Later, when this glycerol 3 phosphate is transformed back into dihydroxyacetone phosphate, the enzyme accepts FAD as the coenzyme. So what is produced is FADH2 into the matrix of the mitochondrion, and it is accepted by Coenzyme Q and then complex 3.
ATP yield from the oxidation of glucose. This is a very important section as far as the examination is concerned. You see, from one molecule of NADH, we get actually 3 molecules of ATP. But as per the latest calculation methods, we get only 2.5 molecules of ATP. That is in the case of the mitochondrial NADH. If we say that 2.5 molecules of ATP are obtained from 1 molecule of NADH, then from 2 molecules of NADH in the mitochondrial matrix, we get 2 into 2.5 is equal to 5 molecules of ATP. Then why in glycolysis 2 NADH molecules, 2 cytosolic NADH molecules are giving rise to 3 or 5? Because from the cytoplasm, these NADH molecules are to be transported into the mitochondrial matrix. When they reach the mitochondrial matrix, they are in the form of FADH2 in some pathways. And 1 molecule of FADH2 will give rise to only 1.5 molecules of ATP. That's how these 2 molecules of FADH2 will give rise to 3 molecules of ATP. And then why is it given 3 or 5? Because in some other pathways, you will see in this same chapter, in some other pathways, the NADH can be transported as NADH itself into the mitochondrial matrix. That's why. Be thorough with this section. Various inhibitors of the electron transport chain, often they are acting as toxins or poisons. You might have heard of cyanide. Cyanide is a respiratory inhibitor. You see where the cyanide is actually acting. It is inhibiting the transfer of the protons from the cytochrome A3 complex towards oxygen, forming water. Also, you can see the action of a site of action of antimycin A and also rotenone. These are actually potent toxins. Some compounds called uncouplers inhibit the phosphorylation of ADP, that means formation of ATP. Okay. At the same time, electron transport is not affected. A good example is 2,4-dinitrophenol. and then antibiotics such as valinomycin, gramicidin, etc. Gramicidin A, etc. are also uncouplers. And how are they acting? It is shown in this note very carefully. Just go through it. Agents that interfere with oxidative phosphorylation. That means formation of ATP by means of the flow of electrons through the electron transport chain. You just go through this list. One or a few of them may come for the examination. Two methods of ATP synthesis. Actually, there are three methods. Two methods are given here. One is substrate level phosphorylation. It is also called direct phosphorylation, direct formation of ATP through the phosphate transfer from a substrate to ADP. You have seen in a glycolysis a compound having 2-phosphate is giving rise to ATP molecule. Can you just imagine which is that reaction? A 3-carbon compound which is containing 2 phosphate units is giving rise to an ATP molecule in glycolysis. Actually, there is only one such a step in the glycolysis in that case. There is one more case, that is the last reaction. A compound, actually it is PEP, carrying a phosphate is also giving rise to ATP. You should be thorough with these stages. That is actually direct ATP formation or substrate level phosphorylation. Then we have the oxidative phosphorylation. Here ATP is formed because of the redox reactions taking place in the mitochondrial chain. Most of the ATP we get through aerobic oxidation are actually by means of oxidative phosphorylation. Here is the mechanism of substrate level phosphorylation. Earlier I told you the case of PEP. This is how it is taking place. Substrate level phosphorylation. I told you in the previous slide that there is one more phosphorylation, that is photophosphorylation taking place during photosynthesis. Can you tell me the name of the organelle in which it is taking place? High energy bonds. As the magnitude of the negative sign increases, energy content also increases. You can see here enol phosphate bond, for example in phosphoenol pyruvate, anhydride bond to carbon, for example in 1,3-bis phosphoglycerate. Actually you can see here the whole list of high energy bonds. Be thorough with it. One or two may come for the examination. High energy compound. Such a topic is there actually in your syllabus. Also called energy rich compounds. These are the compounds in the biological system which upon hydrolysis yield free energy equal to or greater than that of ATP. That means delta G, the free energy change should be greater than 7.3 kilocalorie per mole. Why is a negative sign given there? Because that reaction is actually, is it exothermic or endothermic when the negative sign is given there? Just try to think and find out. But one thing is there, whenever there is the negative sign, the reaction is spontaneous. Okay. Here you see the classification of high energy compounds. Pyrophosphates are there, enol phosphates, acyl phosphates, thiol phosphates, guanidinophosphates, etc. are there. What about the pyrophosphates? A good example of pyrophosphate is ATP. And then acyl phosphates are there, 1,3-bis phosphoglycerates we found in the case of substrate level phosphorylation. And also enol phosphates. This is another class of high energy compound. What is PEP? PEP is phosphoenol pyruvate. Here you can see the enol phosphates. The bond present in this diagram here is enol phosphate bond. It is formed when phosphate group attaches to a hydroxyl group. which is bonded to a carbon atom having double bond. So here you have an enol phosphate bond. The bond is formed when the phosphate is attached to a hard hydroxyl group. This hydroxyl group is attached to a carbon, and the carbon is having a double bonded structure. Guanidophosphates, also called phosphagens. The bond is guanidine phosphate bond. It is formed by the attachment of phosphate to guanidine group. In this picture, there is a phosphate here, and it is formed to or attached to a guanidine group. So a very good example is phosphocreatine, or I should say creatine phosphate. And acyl phosphates, example is 1,3-bis phosphoglycerate. It is acting there in glycolysis. It's formed by the reaction between carboxylic acid group and phosphatic group. You can see the phosphatic group is attached onto a carboxylic carbon here in this diagram. And thiol phosphate. Here high energy phosphate bond is absent. Instead, high energy thioester bond is present. You see here in this diagram, what you see here is acetyl coenzyme A. This is not acting as a source of energy, and the thioester bond is resulting from the reaction between thiol and carboxylic acid group. This is not a phosphate containing compound actually. Phosphate bond is absent here. Yes, you can see here different high energy compounds and the amount of energy released by them when they are releasing energy by means of breaking the bonds. Which one is releasing the highest amount of energy? It is of course PEP. Select high energy compounds from the following. Of course, it is very easy because anything having energy more than that of ATP, anything having energy or anything releasing energy more than that of ATP. So which are those which are given below ATP or which are given above ATP? Just think about it. Phosphorylated compounds and regeneration of ATP. Actually, substrate level phosphorylation, direct transfer of phosphate from catalyzed by kinases, we found it earlier. And oxidative phosphorylation, indirect ATP generation from proton gradient, that also we found earlier. And what's the meaning of this diagram? You can see here, phosphoenol pyruvate is providing ATP. That ATP and 1,3-bis phosphoglycerate is providing ATP. Those two ATPs are utilized for the formation of glucose 6-phosphate, glycerol 3-phosphate, etc. Phosphate potential. It is a measure of the phosphate group transfer potential of a compound. And this diagram was given in the previous slides as well. A few compounds have even higher phosphate transfer potential than ATP. And this can be used to make ATP when they are produced as intermediates. In this diagram, it is given here, PEP is there, 1,3-bis phosphoglycerate is there, of course these two are there. This is actually an explanation to some of the concepts. Energy produced from a reaction can be coupled to another reaction that requires energy to proceed. Transfer of a phosphate group from high energy phosphorylated compounds can activate a substrate or intermediate of an energy requiring reaction. And you can see an example given here. ATP has a higher phosphoryl transfer potential, phosphoryl group transfer potential. You see, ATP when releases energy, it is 7.3 kilocalorie per mole. It is releasing, that's why negative charge. And it has a strong tendency to transfer its terminal phosphorus group to water than does glycerol 3 phosphate. ATP has a higher phosphoryl transfer potential than does glycerol 3 phosphate. So what do we mean by saying that ATP has more phosphoryl transfer potential? It's very clear from here. This is a basic idea about this unit, this chapter. Gibbs free energy. Actually, you should have seen this slide in the beginning. Gibbs free energy G expresses the amount of energy capable of doing work during a reaction. When the reaction proceeds with the release of free energy, the free energy change delta G has a negative value, and the reaction is said to be exergonic. So actually, in a slide I had asked you why there is a negative value given here. This is the answer for that question. In endergonic reactions, the system gains free energy and the delta G is positive. Remember, whenever the delta G is positive, the reaction is not spontaneous because we need to supply energy. At the same time, when the delta G value has a negative sign, the reaction can take place by itself. We say it is a spontaneous reaction. And when the delta G is negative, it proceeds forward. When it is 0, reaction is at equilibrium. When the delta G is positive, it is reversible.